Speaker is Rosemary Gillespie, and obviously Rosie really requires no introduction here. She's a professor in ESPM, obviously, a very well-known member of the community here, and she's going to be telling us about her work on adaptive radiation in spiders.
Thank you, Jim.
Thanks, Rosie.
Yes, I should say this is very intimidating. You know, to give a talk to people when you know everyone in the audience, it's extraordinarily intimidating. But what I want to do today is just talk to you a little bit about some of the work that we've been doing recently, and kind of give you a feel for where we're going with some of this. I'll be covering a lot of it fairly superficially, and you can always ask questions at the end. And some of it I'll just be touching on almost not at all, but just to show you the direction we're going in. So the main focus then is looking at the origin and predictability of diversity on oceanic islands. And so in this vein, I'm going to talk about two main things. The first, I'm going to talk about diversification, and so how species diversify, their ecological shifts, evolutionary convergence, how the fauna build up, and the role of plasticity, and what kind of plasticity as well. And then I'll talk about the aspects of variation upon which selection can act. So, looking then at diversification, and what I'm going to do throughout this talk is focus on the Hawaiian Islands. And why do we focus on the Hawaiian Islands? Most of our lab do focus not on the Hawaiian Islands, not all I should say, but most of them focus on remote islands in the middle of the Pacific. And the reason for doing that is that these islands are, they're really isolated, and they're just very discrete areas within which you can see kind of just a packet of evolution and in an ecological context. And if you can't see Hawaii in that massive ocean, there it is there. And the islands of Polynesia, which I won't be talking about, are down here to the islands of Tahiti and the Marquesas are down there to the south. So the nice thing particularly about Hawaii is that it is a hot spot. And so that means that it comes from an underwelling where the lava oozes up. And as the lava oozes up, then the plate moves up to the northwest, and as it does so, as the plate moves away from the hot spot, the islands become eroded, and eventually they go down to nothing. I should say that there's the current high islands are all considered high. They change, but they're all they vary with they don't go gradually down. There's a big gap between Kauai and the northwest Hawaiian Islands are simply atolls. But the nice thing about the current high islands is that they provide this chronological arrangement where you can actually see stages of evolution as you go across the island system, or across the different islands. So here you've got the islands, and what you would expect if an organism has been in the chain for at least the 5 million year period, at least the period of the oldest island, then you would expect that organisms would simply hop down the island chain from the older to the younger islands. And so this is called the progression rule, where they simply, you know, inhabit, they hop down to new islands as they appear. And some taxa have done this really well, where they've just simply hopped down the island chain. This is Orson Welles. It's a lineaphiid, a sheepweb spider that Gustavo Hormiga described. Gustavo liked Orson Welles and named them all after Orson Welles plays, but whatever. Gustavo, with me and Mikel Arnedo, we developed lots of molecular markers and did a phylogeny for the group across the Hawaiian Islands. And this is a while ago, but what's nice about it, and again, I'm not going to go into detail about what what we did, but the point is that what you find here is that you get a high diversity on the older islands, and then they basically hop down the island chain and form new species as they go down the island chain. What's particularly interesting, though, about this is that it's a non-adaptive radiation. They all basically do the same thing. And So that's all fine. You just get this organism changing basically in allopatry. But what I'm going to concentrate on is groups that have not only hopped down the island chain, but have also diversified within the archipelago, often within islands. And when you think about this, you think of the kind of classic situation of Hawaiian Drosophila, known for its rampant proliferation within the Hawaiian Islands. In Drosophila, many of the major ecological shifts have occurred fairly low down in the radiation, and speciation is largely associated with founder events or shifts in sexual behavior. I don't know if this is going to, I can never tell whether these are going to work. Here we go. So these things have very elaborate sexual behavior. This is on the right is a male dumping pheromone on the head of the female and sticking his tongue out and all of this kind of things that flies do. And, so it's a very elaborate display, and speciation is associated with modifications in this sexual behavior and occurs very, very rapidly. So you get, very quickly, you get speciation happening. And even on the youngest island, you've got pretty much full-blown speciation. So what I'm going to talk about, let's see if we've just decided to stop here for a second. Let's just sec. Yeah, maybe this will work. Yeah. So what I'm going to focus on is spiders, and I'm going to give you a couple of stories here about the different spider groups. And the first one I'm going to tell you about is one that I really love. Well, actually, I love all of this. This one is, these are stick spiders, and this just shows you kind of a spectrum of the different ones. And what's really cool is that some show this matte color, some are shiny, some are brown, some are really almost black, some are short, some are long bodied. What we've done is we've, I described most of them fairly recently, but we've been working on developing a phylogeny. This is just a very basic majority rule tree that I'm just showing here. But again, the point is, and I haven't actually got the names even there, but the point is here that what they do is they hop down the island chain. So they've been in the islands probably somewhat older than Oahu, not quite as old as Kauai, but they do show the hopping down the island chain. And within this context, you find all of this diversity. So where do all of these colors and things fall within that framework? What you find, so these are just the islands, they hop down the island chain really quite nicely, and so you see that, so you see that the Kauai is up here. It's fairly poorly resolved, and we're still working on that. But it's you get Kauai, Oahu, then the Maui Nui complex, and then the Big Island. And what's interesting, though, is if you look at this in the context of the color morphs, so what you find is that these gold ones pop up all over the tree. And likewise, when you look at the brown ones, they also pop up all over the tree. So the brown ones and the gold ones have evolved repeatedly. What we're finding also is these white ones associated with white lichen in mesic forest, they also pop up repeatedly because we've just found one on Oahu. So you're getting this convergence of form in across this group. And this is one that we're still, Andy might be working on, Andy Rominger might be working on a bit more. But I'm going to leave that one now and talk to you a little bit more about one that I've been focusing on most extensively. And this is the genus Tetragnatha, these long-jawed spiders. This group is one that you can find the genus anywhere. They're just, if you go down to the creek, you'll find them in the creek. They're up in Tilden, they're all over the place. And when you find these spiders, they're just like this. They're long and skinny, and they build very flimsy webs. You can't even see the web. It's just a very flimsy web over water. So very homogeneous. And then the thing is, you come to Hawaii and you find this extraordinary diversity of form. They've just basically gone wild. They've gone into all sorts of different habitats. Some are web building, some are not web building. If you go into a given habitat, you find round about 5 to 10 species in any one site. So just to show you, and if I can get this one to play here, just to show you a little bit about what's involved, you have to go out at night. And so these things, just in the daytime, it's almost impossible to see them. But at night, that you can just go and pick them up. There we are, just rolling and so they tell you exactly where they want to live, how they want to live. So they just sit there, and you can measure the web structure. They tell you what kind of prey they like to feed on, and so you can watch them feeding, and you can watch where they like to feed, what they like to feed on. The only downside is, of course, that you have to work at night. So they're a really cool group to work on. This is a spiny leg one. This is one that does not build a web, and so you can just watch them running around. They won't. It really doesn't affect their behavior if you just sit and watch what they're doing, and you can see they'll just run between the branches and basically tell you what they're. Not quite what they're thinking about, but and this is Darka Kutaris demonstrating how he's catching the spiders. This is a mature male that's in pursuit of a female, and this is one thing that Susan Kennedy and others in the lab might be looking at, just how they're actually finding who they're going to mate with. Also, how the form of the web differs in different sites that they're going to be that they live in. But we'll talk more about that later on. So they're a great group to work in, and they're very, very diverse in this in these habitats. So so the first story I want to tell you within this group is how the web builders have diversified. So the web builders are, as I said, they're very diverse. But they all build the same kind of web. It's still a flimsy orb web. It's not over water anymore. They're just diverse within the forest. But Todd Blackledge, who was a postdoc in the lab, he made lots of measurements of the different webs that these spiders build. And he found that in a given area, they'll spin specific kinds of webs. So he did all of these measurements. He also measured the kinds of prey associated with different species and the different webs. And the point here is really just, what you can see here is that this is the Eurychasma, which I used to call big step because there's big steps between the spiral lines. This is Feliciphilia that's under ferns. These ones all live together, but you can see there's a very different prey composition that they're getting due to presumably the different kinds of web that they're building and the different place that they're building it in. So what Todd did then was he did a principal components analysis of these web measurements and found that most of the differences were explained by silk density and web size. And so when you looked at a given site, you found, so this is one site on Oahu, this is one site on Maui. And so what you find, based on the principal components analysis, the 3 different species that you get at that site really occupied very different parts of the ecological arena based on the silk density and web size measurements. So likewise, they were well separated on Maui and likewise Hawaii. A lot of overlap when you put them all together, but the bottom line is within an area, within a given site, they're well separated. And so what he did then was look at this in a phylogenetic context, and again, this is a very, very basic phylogeny for this group. But this is just what he published in PNAS a while ago. But the point here is that the ones with similar web types, this is Hawaiiensis and Stella robusta, that spin very similar web types, but they're not closely related at all. Likewise, Feliciphilia and this one on Oahu that I haven't described yet, and Emerald Ovoid that I still haven't described yet on Oahu, and Eurychasma, they spin very similar web types, but they're totally unrelated. So what you find then, I mean, the take-home message is that you get evolutionary convergence of web ecomorphs. They're converging to the same form of web. What's actually really interesting here as well is that, you know, you're getting a story of lots of convergence here. But if you look at the occupants of those webs, so this is the tight line, and these ones, these two, the occupants, they look quite similar. But look at these ones with the loose webs. They're just poles apart. This is a big, fat, green one, and this is a kind of maroony, nondescript one. And the occupants are morphologically all over the place. So there's no evidence of convergence of the morphology, but lots in terms of the web form. So the group, though, that I've done the most work on in this, in the Tetragnatha are these spiny-legged ones, and I'm going to talk to you a bit about them, because the spiny-legged species, the ones that don't spin webs, they occur as three distinct forms. Sorry, as four distinct forms. So there's a green one, bright green, and this one lives under leaves. There's one, there's a large brown one that tends to live on tree trunks, a maroonish one that tends to live in moss and lichen, and then a little brown one that tends to live on twigs. So Becca Carter, who is a graduate student in the lab, she looked at how these species are partitioning the habitat, and I'm going to go through this just very quickly. The bottom line here is that in terms of the niche breadth, these are just, we're just looking at green species across the different islands. So this is the different greens on all the different islands. And you can see that they do very similar things. The orientation is just, this is just looking at the niche breadth. And it's showing that they have very similar niche breadth, and it's a very narrow one. Basically, they just live under leaves, under green leaves. And also, they in terms of the substrate type, they tend to live just, again, under leaves. And so that's really all that they do. They just sit under leaves. I won't worry about that one. Also, you get strong similarities looking at the green species across different islands. They basically do the same thing. This is just looking at the niche overlap across different species on different islands. And you see that the overlap there, but represented by these high numbers, is really astounding. They basically do the same thing. They live the same lives. Then if we look here, that within a given community, this is at one site on Maui that she looked at. And the things to look at here is that the green, this is Tetragnatha waikamoi, comparing the green ones to the brown ones, the green ones to the maroon ones, what you find is there's a lot of divergence in all of these attributes when you look at the overlap. In other words, the green versus brown, green versus maroon, there's little overlap in the kinds of habitats that they occupy. Just also to mention that the Kamako, the maroon one, is divergent from the big brown one in terms of the substrate taxon, the kinds of substrates that they live on, representing that the maroon lives kind of on mosses and lichens, whereas the big brown lives on tree trunks. And Quasimodo and Kamako are quite similar in some aspects of the kind of habitat that they live in. But the bottom line is that even, here's Brevignatha and large brown on another site. And just again showing that you get the large, very little overlap in the kinds of habitats that they occupy. This also, I should say, reflects in the kinds of diets that they have. So these are the green ones. This is based on a very small sample size, but you can see that there's a lot of these little homopterans that you find under leaves in the diet of the green ones. This is across different species on different islands. And then in the maroon and the big brown ones, they tend to feed on things that you get more on tree trunks. In particular, Lepidoptera larvae, or caterpillars, they feature largely in the diet of both these. So the bottom line is you get big differences in the kind of habitats that these different ecomorphs live in. And I should say, and I'm just going to mention this because this is the work of Pete Croucher, who's a postdoc in the lab. And Pete has been looking at the microbes associated with these species in the different sites. And what he's found, and again, emphasize that this is very preliminary, this is just a heat map of bacteria, bacterial relative abundance that he's developed. And what you're actually seeing here, these are, you can't see these, but these are different microbes that he's picked up through transcriptome analysis. And what it shows here, these are here you've got the maroon ecomorph and the green ecomorph. So what this heat map does is it groups together things that are most similar. And it's showing that the maroon and the green are more similar to each other. And I should say this is looking at the whole animal, so it's picking up everything on the outside as well as the inside. But what he also did was look at the venom, which is just going to be looking at the inside of the animal, and found that when you look just at the inside, the bacterial or the microbial assemblages separate out according to island. So you get distinct groups according to island. There's a lot more work to be done there, and Pete can give you an update when it comes out. But the main point that I want to make here is that there's some evidence, although preliminary, that the maroon ones have their own microbial composition as compared to the green ones. And this seems to be reflecting the different kinds of microenvironments that they live on. So you've now then got a good feel that these things live in different environments, but let's just look at how they've actually evolved. So you see, what I've done here is just look at the species in the different sites. So this, for example, on Oahu, you get a green, a maroon, a big brown, and a little brown in this one area. And in most islands, in most habitats, you get one of each. So the question then is, how did they evolve? And if you do, this is a phylogeny based on multiple markers. Mitochondrial DNA, Alzheimer's mini satellites, and we're getting a bunch more at the moment through the work of Dr. Kataris. And so the bottom line here is that what we've found is that they tend to hop down the island chain. So the most ancestral is on Kauai. So they show a pattern of hopping down the island chain. But the most interesting thing is that so this just shows the going down the island chain. But where you see, here I've coded them according to ecomorph, and you see the green ones pop out over and over again. This is the brown one, and it pops up over and over again. This is the maroon one, and it pops up over and over again. And so they're again here, what you're seeing is repeated evolution of the same form. However, you also get ones that evolve and then simply hop down the island chain. So this is a big brown one that evolved somewhere in the middle of the islands and hopped down the island chain. And likewise, the little brown ones, they do the same thing. So you say, that's a bit of a mess. So how do these ecomorphs arise? I mean, what are we trying to say here? You like to have a nice story. So the thing is, it seems that how I would say it is, it's like a race. Who can fill the ecological space, so to speak, first? So can you fill that space first by dispersing into it? Or do you just hop down the island chain and fill it more quickly by in situ evolution from one ecomorph into another when you get to the new island? And it seems like there's both going on, and it's a race as to who can do what first. Okay, so the thing is then that you've clearly got huge diversification originating from apparent homogeneity. And you get repeated evolution of similar forms. And I want to take just a little detour here and tell you how this can actually occur at another level as well. And I'm going to just quickly here tell you another situation where you get the same kind of convergence, but occurring, the convergence resulting in more diversity. And you'll see what I mean in a second. So what we found, this is in another group of spiders, and what we found is that certain taxa show similar patterns of within species diversity and repeated evolution of similar within species diversity. So let's just look at this. This is the story of the happy face spider. So it's a very cute animal, but it shows genetically controlled color polymorphism. And you think, well, gosh, I'd love to go to Hawaii and look at this spider. You know, it's so adorable. But then what's interesting here is that you say, well, yeah, it is really, really adorable. But you actually get a very similar one in Tahiti. And you actually get a very similar one in Micronesia. And this is in Fiji. Look at that little dot in its butt. And, I mean, they show this similar color polymorphism across just about every habitat. This is pictures that Darko Kataris just took from the Juan Fernandez Islands, and you see very similar color polymorphism, just like the happy face spider. And then, you know, what do you find? You look in your backyard and you find the same thing. This is Theridion californicum. You can just go up into Tilden and find Theridion californicum, and they match almost 1-to-1 with the happy face spiders. So you could argue which is cuter, but I mean, it's really remarkable that they show a really similar parallel evolution of the same kind of diversity of form. So these equivalent forms in Theridion californicum and Theridion gradator across these very different places. In terms of how this color polymorphism evolved, this is a very basic phylogeny, and again, Darko's doing much more on this. But the point here that I want to show is that these ones that show the color polymorphism pop out all over the tree. This is the subfamily Theridiinae, which is a bit of a mess. But the real clincher here is that the evolution of this color polymorphism has occurred repeatedly. But what we've been focusing on, though, is the Hawaiian happy face spider. And this is really an interesting story, and I'll tell you why. So the happy face spider then occurs on all of the islands except Kauai. And in every habitat, it exhibits multiple forms. And I should say that the most common form is this one that's plain yellow. The plain yellow is very hard to see. It almost disappears against the underside of the leaf. Then you get all of the rest of the population, which is about 1/3 of the population, all of these other different colors. So all of these array of colors, so red the front and back, sides, ring, blob, white. I mean, a tremendous array of colors. So what we found, this is just to show you the array, why do you get such diversity? And this is something we've focused on quite a bit. So, as I said, the frequencies of the color morphs, the yellow to the pattern, so this one to all of the rest, is almost identical in the different populations. And what you find is that what we wanted to see is, you know, why they're so similar. And so the first explanation will be that they're just moving around a lot, which would be makes sense. I mean, the islands aren't that far apart, and so they're just moving across the islands. So the other explanation is that there's strong selection maintaining the frequency of this color polymorphism in each population. So again, Pete Croucher did this work with looking at AFLPs, and well, actually this is mostly from Alzheimer's, but this is a structure plot just showing the separation of the different populations. So basically the different colors represent different populations. And what you can see, this is Oahu, Molokai, West Maui, East Maui, and different populations on Hawaii. And you can see each population is really remarkably distinct. The only exception is really this place here, Kilauea, which is just so young, it's only about 300 years old. So it's not surprising that it's drawing from multiple populations. So the bottom line here is that there is very little movement between populations. So they're just, they're really not moving at all. So what can we say? Based on this genetic analysis, there is not movement between populations, and so there's got to be selection acting on the color polymorphism. And what is the selection? It's got to be, the most likely thing anyway are these vicious little animals. But honey creepers actually really do a number on the spiders, and they're a strong selective force. And so it seems to be that their ability to develop a search image, and so they're selecting, they can select for the most cryptic form. And if the most cryptic form builds up to a very high frequency, then there will be strong selection to be anything but the most cryptic form, just for diversity per se. So what we did then was this is work I did with Jeff Oxford, and what we did was we did lots of crosses between individuals initially on Maui, just crossing yellow red front, all of these different colors. I'm demonstrating these crosses because they're simple, and what it's showing are the situations where the colored ones are heterozygous, and the most usual form that they're heterozygous for is yellow. So yellow is bottom recessive, recessive to everything else. So when you do these crosses, they fall out one-to-one, the colored and the yellow. So this is all very straightforward, and then we came over to the Big Island because it's great fun to do these experiments. And so Jeff did the same thing on the Big Island of Hawaii, crossed a yellow and a red front. Half came out red front, half yellow. What's interesting here is that the red front are all male, the yellow are all female. And when this is just quite baffling. And then you cross a male yellow from Maui, because we don't get male yellows on the Big Island, with a female yellow from Hawaii, you get the red front characteristic of the males in Hawaii coming out in the F1. So what in the world has gone on here? We still don't know, but it seems that, I mean, in terms of the big picture, what seems to have happened is you've got colonization from Maui, which is now fairly clear. With that colonization, presumably just a couple of individuals would have colonized, and then you would have got strong selection from these initial colonists to recover the full spectrum of diversity of the parent population. So presumably there was strong selection to get back to what they originally were, and they did that through a different genetic mechanism. So here you've got convergence to the same array of color morphs within a species. And I'm not going to say much about this because it's very much a work in progress, and again I want Pete to talk about this himself, but Pete Croucher has been working on this problem for the last few years, doing next generation sequencing to try and figure out exactly what the genetic basis is for as they've gone from one island to the next, and to try and figure out what genes might be involved as they've gone between islands, and how selection has acted on these. So that's what I want to do now, though, is so it's enough of the diversity between species, diversity within species. What I want to do is just look a little bit now at where selection is acting. And you've got between species diversity, and what I want to look at is how the diversity arises. We found that diversity between individuals within a population doesn't really lead to much in terms of adaptive differentiation between species. And so what I want to focus on is more within an individual diversity. And this is environmentally induced, food-induced, physiological, developmental, and how these might feed into between species diversity. So what we're going to look at then is translating within species variability to between species diversity. So the point is, huge diversification originates from parent homogeneity, how? And repeated evolution of similar forms, why? This is work that was started by Mary Jane West Everhart. She did a really important series of papers and wrote a book about this. But the main point is that, contrary to common belief, environmentally initiated novelties may have greater evolutionary potential than mutationally induced ones. So basically what she was arguing for was plasticity and variability within individuals, and that selection might act on those. So looking then at the spiders, I'll just mention this and not go into it. Spiders can go through various different kinds of change within individuals. I think everyone's familiar with crab spiders that can change according to the substrate that they live on, and I won't worry about that, but they can change if they go onto different colored flowers. Happy face spiders, if they eat something different colored, they can change their background color. Spiders also have the ability to undergo physiological change. This is one on Molokai, and this one, if you prod it too much, it'll just change to this very dark color. They cover the guanocytes, and this is a defense mechanism so that you can't see them. But there's also developmental switches that they can go through. And this is the Tetragnatha hawaiiensis, and this is work that Becca Carter did in my lab. And what she found, here's Tetragnatha hawaiiensis. this is the same individual just a day later. So it changed radically. So it's, and what's interesting here is that this species belongs to a lineage that's radiated. So the thing is then, why is this going, what's the role of this? Does it have a role? So here we look at other radiations. And so if we look at other radiations, we know of, in particular, the famous situation of anolis lizards. You get repeated evolution of the same form. You get these same kinds of repeated evolution of similar form actually across multiple different groups. Perhaps the best known, besides the anolis, are different lineages of fish, and in particular the cichlids of the Great Lakes of Africa, where you get the repeated evolution of similar forms between different lakes. You also get this same kind of thing going on in sticklebacks, where you get repeated evolution of limnetic and benthic form in sticklebacks, and this is Dolph Schluter's very famous work. So these studies suggest, because they're so similar, the forms that you get, they suggest that there's a similar genetic basis for this. And there's a very exciting thought there that there's a role of plasticity. So what is that role? And again, here we go to Mary Jane West Eberhard's work, where she argued that the pattern of ancestral plasticity can determine which behavioral phenotypes are expressed in a given environment, and therefore which phenotypes have the potential to evolve in response to selection. So what she was arguing was that ancestral plasticity at the onset of adaptive radiation can actually guide subsequent adaptive change. And so people have now looked at, first in sticklebacks. This is what she showed. In sticklebacks, individuals of anadromous species are limnetic when young, and they become more benthic with age. In the sticklebacks, this is recent work, that's shown that in basal species of these cichlids, basal species experience developmental shifts in genes associated with photoreceptor sensitivity, and the more derived taxa have undergone parallel shifts to opsin genes sensitive to either short or middle wavelengths. So ancestral plasticity translates to fixed differences. So let's look at the spiders. And here, well, you say, well, where do you find it in this whole story? But if you look here, what you find is on Kauai, the green one is missing. So this is the one that Becca was looking at. So this is the one where the green actually shifts to this maroon form. On the younger islands, they're either maroon, little brown, big brown, or green. They don't switch. They've made up their mind. So the plasticity, or the heterochrony, is limited to the older islands. So this again is the work of Becca Carter, and I won't go into this in detail at all, but what she did was she scored the colors of these spiders, the ones that changed color and the ones that didn't change color. And what she found was she did this across, first of all, looking at age since maturity. And what she found was they started off green. This is a principal components analysis, but with the main principal components summarized by the maroon coloration. So if you plot that against the age, what you find is more and more maroonness as they get older. So there's variability on the exact time, but they almost all end up maroon by the time they've reached their final stage. If you look across the islands, you get, this is the degree of maroon coloration, again represented by principal components analysis. And here's the amount, just comparing the different supposedly green species on the older islands. So this is Kauaiensis, this is Polychromata. And these ones here, these are Waikamo and Brevignatha, and they don't change at all. So basically what she was arguing is that you get progressively more juvenile appearance of the climax state of mature individuals of more derived species on each island. So it seems that it's a kind of heterochronic shift where the green form becomes mature on the younger islands. So if you look then in a phylogenetic context across the islands, what you find is that these ones that shift color are on the older islands then. And then you get just one, this one does not, and this one does not, and then none of these shift color. So you get this color shifting appears to have been completely lost round about somewhere on Oahu and leaving Oahu. And it's given way to species that just simply never change color. So what we're trying to do now is just figure out how this has happened. Whether you get mutations in the same pigment pathway or regulator gene, activation of different pigment genes, or changes during development. So are these just simply heterochronic shifts in pigment pathway or regulator gene expression between the ones that change and the ones that don't change color? And I'm just going to mention here, again, the big thing is yet to come, but Michael Brewer, who's a postdoc in the lab, he's been working on this to try and figure out whether selection has been acting on these genes. And what he's found, intriguingly, but totally non-statistically, is that a few of the genes that show signatures of selection are also associated with development. And this is a tremendously exciting result, but we have to show that it's real. But it'd be wonderful to actually be able to show that it's just a developmental shift that's happening here. So, okay, so we've got here, what we're seeing is that at least in this radiation, is it between individual variation that's important or within individual variation that's important in really allowing the exploration of ecological space that the spiders show? And what we've clearly shown is that it's actually, the big factor is the within individual variation that seems to be playing a really huge role in this. And this is what we're looking at the moment. So just before I finish up here, you've got, I'm just going to say that what we've shown is rampant but not complete convergence to similar phenotypes on different islands. Niches are filled by an interplay between colonization of species from one island to the next, or by adaptive shifts from another niche on the same island. And then when an ecomorph is missing, you find this developmental plasticity, such that a single species can occur as two different ecomorphs. And this seems to play a big role in facilitating adaptive radiation. So I just wanted to mention finally what we're doing now. We've got this big project that we've started just to try and figure out why, basically how diversification is happening. So what we're doing is we're looking across the islands basically to see what the different stages are as organisms just start to diversify and how they accumulate and how they go extinct. I should say that this work builds on patterns that we found that I'm not going to go into at all, but basically the patterns show that in some very large groups you get a peak of diversity in islands of middle age. In smaller groups they just keep accumulating, and this is both for plants and animals. This is work that I did with Bruce Baldwin. There's also a well-known pattern in the vegetation literature where fairly early on in the island succession you get a peak in nutrient availability, and so that dies off, that kind of drops down on older islands. Peter Vitusek has been building on this work a lot, and he's been looking at patterns of changes in canopy nitrogen and other features as you go across this age structure of the Hawaiian Islands. This is starting on these are 3 sites across the island of Hawaii, and then Kauai, the oldest island, and you see these major changes in the patterns of canopy nitrogen as you go across the islands. So these are these intriguing differences across the islands. So what we want to do then is look, yes, across all the islands, but really where it's all happening is on these very young islands. The big island, when you look at species there, we always say, oh, they're populations from Maui, or they've just started diversifying. To be honest, having done all the taxonomy of the organisms there, it's a big mess, and it's really hard to figure out what's going on. And this is really what initiated the work that is designed to look at how diversification is actually happening, and the role of mixing and separating in fostering diversification. So what we're focusing on then is the big island of Hawaii. Here you see the substrate ages, and what we can do is just look at the association between substrate age and diversification, and look across multiple groups to see how the community comes together. So this is work that I should say John Hart and Andy Rominger have been instrumental in this, and they're going to play an instrumental role here. But one of the things that we're going to do is use the maximum entropy theory in ecology premise to try and understand patterns of species area abundance at a given site. The basic idea is that energy in a given site is packaged most parsimoniously through individuals and to species, and there's a prediction based on this that given energy in a site, you'll get a certain species abundance at that site, and you can use this. It's a prediction based on very little biology. But the thing about it is that it gives you something to actually look at. So if you see, these are just based on data that Dan Gruner, who's part of this project as well, so Dan accumulated these abundance data for arthropods across different islands, and different sites of different age. And what Andy, working with John, found that when you looked at these species abundances in the context of the maxent predictions, you got very close matches, except at certain sites, in particular those kind of intermediate age. And Andy might want to say more about this at some point, but these are very preliminary predictions, very preliminary assessments. At the moment, we're going to get much more data to try and figure out exactly what's going on, and whether it's detritivores that show this pattern, whether predators, whether it differs across different trophic groups. But what you want to do is just see whether you can tell something about how diversification is happening, whether there's too few species and they're going to fill the vacuum in sites that deviate from the maxent predictions. Also, part of the project also is Neo Martinez. Neo does food web studies, and this again is something Andy put together to try and portray what we're trying to do. But the kind of idea is that you've got very simple, very basic food webs on these very young sites, and over time they become more complex. So can you predict the little balls that are going to come in and fill these little gaps here? And this is what we're going to try and see. So basically trying to see what fills these little holes here. And at the same time, try and see how these filling of the little holes affects the diversification of taxa across these different sites. So it's basically putting together the ecological changes with the evolutionary changes. So that's it in a nutshell, and I just wanted to thank everyone that's been involved in this. Ev Schlinger has provided much of the funding for this, and our graduate students, Andy and Darko in particular, but also Becca, whose work I talked about a lot, and Athena, and Michael and Pete, who you'll see at the back of the room somewhere, and lots of people in Hawaii who I've worked with very, very closely for many years, and my two very close colleagues, Jeff Oxford and George Roderick. And thank you for having me, and I'd be delighted to answer questions. That was a lot to cram into the time.
So we'll give people just a moment that have to go to their next event a chance to walk out, and then we'll take questions.
Could you get the light on the way out?
Shall we? Questions for Rosie?
So I think this idea that as things colonize, or the older islands, they sort of lose plasticity and become genetically fixed for various ecomorphs is really interesting. And then so I'm wondering, kind of from a colonization of the islands initially point of view, do you think that the initial colonists are therefore the plastic ones, or because presumably they're coming from places where they're very old, So why would you start off?
That's a really, really interesting question. I don't think they're plastic themselves. However, I mean, we've got a species in California that's very closely related to the bottom of the tree, and that the California tetragnatha are that they're all fairly well characterized. So we've got a fairly good handle on this, but Darko's going to be exploring that further. But the point though is that these ones so all the closest ancestors at the moment are just blah, elongate brown. However, if you look at tetragnatha across the world, here, there, and everywhere, just a bright green one pops up. And so there's a bright green one on the east coast of the US with long leg spines, runs on leaves, it's away from water. There's one in India, there's one in Japan, there's one in Australia. So they just, it seems that they have this in them. And so presumably, even these boring old ones down on the creek, they have it in them. but exactly how it gets to where it's expressed, that's the thing that's going to be really interesting.
It does seem like they would be the best colonists of a new island would be the ones that do have some potential in them like that.
Right.
In the sense that they would.
Well, the thing is, it's not surprising that this group has colonized the islands, because they live right by water, including the ocean, and they're super abundant in salt marshes and things. And so if you sample, they've done sampling of the aerial plankton over the oceans, and you get gobs of these things. So it's not surprising that this group itself has radiated. It's just that, given that presumably that the taxon itself that got there was in all probability one of the blah brown ones, then how did it start to display, the potential for all of this diversity? I mean, to what extent was just the environment there and the small population size or whatever other situations, what about that allowed them to display all these different variable morphologies? And the other interesting thing I should say, the other interesting thing is, why do the ones in the older islands show the plasticity, the heterochronic shift? They've been sitting there for just as long as the other ones, and so why have they not evolved? Because what I've argued is, well, that the green ones and the green ones stick to green leaves. The ones that shift, they may not be quite as good at it as the green ones or the maroon ones. It seems that it's quite difficult for them to choose substrate. They can't see or anything. So you say, well, you can see the selection as to why they'd fix on either being green or maroon, but why would you maintain this ability to shift? And again, I don't know why they would do that, unless there's something about the colonization event itself that allows some kind of genetic movement and diversification. Yes, Ellen.
Do you have any information on population sizes of these different populations?
Oh, that's an interesting question. Pete, do we have any idea? No.
Variable, depending on what you get. The tetragnatha probably a lot.
Yeah, I suppose we haven't actually. Yeah, we haven't really estimated. Some of them are going to be tiny, especially that one of the things that we've been doing is looking at an idea that Hamp Carson had, and Hamp Carson, you probably know all of this, but he got a lot of flack for some of the ideas that he brought up, because they were entirely speculative and all about these, how the genetic changes happen, and that you get all of these epistatic things going on. But we're now at a position where we can actually look at that, and I think there's actually more in what he talked about than people have given him credit for. But what we've been doing is looking at differences in populations between these little islands of vegetation on the Big Island where you get the lava flow has fragmented the habitats. And what you find is if you look between genetic differences between these little islands of vegetation, you find really remarkable genetic differences between these kapukas. And you say, you know, they're a stone throw apart. And you say, well, and they're not that old because the lava only flowed, whatever, less than 100 years ago. And so you say, well, how do you get genetic differences between sites that are just a stone throw away from each other in such a short time? And the thing is, what it seems, you can't get divergence in that time, so it's got to be some kind of recolonization. And presumably you get mixture then going on. So you get colonization of these habitats, you know. Presumably the lava flows around them. Presumably also it gets really hot and a lot of them die. And so presumably also it would provide opportunity for others to hop in there. And it seems that based on the what work we've done across this area of kapukas, there's tremendous genetic diversity contained at this site, and it seems that this is playing a role. This kind of admixture is playing a role in fostering genetic diversity. But exactly what its role is in fostering divergence at the same time, we really don't know, because they can't last long enough to allow divergence to happen. They'll regrow. Yes, Damien.
Hi. Is there any difference between either the dispersal abilities of the ones that can shift color and the ones that don't, or between the ones on older islands and newer islands? Do the newer islands just stay put more?
That's an interesting thing. So there are major differences in how much they disperse. Overall, these organisms disperse really remarkably little. So you get ones that are found, you know, this one that I showed that's lurid, fat, lurid green one that is just found on the summit of Oahu. And you go down, you know, you go down the slope of the mountain, it's not there. And you go to the other mountain, it's not there. It's just at that one site. So they're, you know, by and large, they're very restricted in how much they move. So that said, there are some that move around a good deal more, but there are just a few of those, like Tetragnatha quasimodo, the humpback one, but it in terms of the coloration, Quasimodo, Darko's working on Quasimodo, and the coloration is, you know, I always thought it was just this big fat brown thing, and it's actually very variable in color, and Dark is currently documenting that. So I can't think of any role of color related to dispersal, but I hadn't actually thought of that. But I don't think. Can you think of any role of how it could? I don't think so, Damien.
Well, I think about in these lizards and things where the melanins and so on really have big effects on behavior. Is there any possibility there's pigments in these spiders that also have pleiotropic effects on behavior?
Well, that certainly. I mean, so the green ones do. You know, they do all of these other things that was shown. I mean, they live in different places, but they're much more agile. They have these, you know, all of the spiny ones have long leg spines. The green ones have very long leg spines. When you get when you get the green ones popping up all over the other parts of the world, the green ones also always have these long leg spines. So it can't be, there's got to be some association there. It can't be just random that they're doing this. But exactly how they're tied, I don't know. Melanin, I don't think you haven't found melanin at all per se, melanin per se, but Pete's looking at all the genes for the different colors. But nothing like the melanin in the lizards. Yes?
Can I just ask one last question?
Yes.
So you talked about the green ones that are cryptic, and then some of them there's a polymorphism there where some of them have the red happy face, and that was like a deviation from the the cryptic pattern. Is there any reason to believe that aposematism could play a role in this, and there could be some tension between being cryptic and being aposematic? Or are they not dangerous enough to have an aposematic coloration?
So they're not at all dangerous, unfortunately, for them. So
they're not venomous?
They have venom. The birds like to eat them a lot. They have venom. But so the happy face spider is in the family Theridiidae. The long-jawed spiders are tetragnathids. Both those families are very. There's no indication that if a bird could see those spiders, it's going to eat them. What Jeff has argued is that the reason you get the diversity of color is that birds have this, building on the apostatic selection argument, but birds tend to avoid novelty and in their diet, and so they'll tend to. I shouldn't be saying anything about birds.
Is there anything else in Hawaii that might have red spots that is toxic that they could be mimicking?
Well, they mimic. There's a lot of things that have red spots. In fact, anything that lives under leaves has. So it's really interesting. So this is off topic a little bit, but you see what's really cool is I showed that these stick spiders that live under leaves are gold. The tetragnatha that live under leaves they're green. The happy face spiders are translucent yellow. All of them have a red color polymorphism. They all have blotches of red variable across the population. So presumably birds are responsible for all of these.
If you looked at them under UV?
No, that'd be cool, wouldn't it? It'd be great to see.
Because that might have a hidden. So some of the bird work showing that, like blue tits will preferentially find caterpillars based on the UV reflectance rather than the actual coloration.
But they're not trying to encourage the birds.
And then that reflects the extent of their nitrogen content.
So presumably this is for avoidance, though, Rory, right?
Right, but I was just thinking, maybe those green ones actually have those patches which reflect UV, and then there's a switch off with that, and so it gets expressed as another color. So the expression and the color might be decoupled. So it might have that pattern, it's cryptic, and then that gets turned off for whatever reason, and it's just
or it could be changed to a different pigment that's reflecting in a longer wavelength.
So it's less cued in upon. If it's driven from a bird visual perspective.
Are there things on the bottoms of leaves, like galls or things like that are inedible?
Yes, and that's
that they might be mimicking.
So yes. So when they show this color polymorphism, it seems. I mean, the other thing is that when you look under a leaf, there are these dots. There's dots from fungi, from the galls, from all sorts of things, and so. They're doing. It's either that they're just being something different, or that they're trying to break up the pattern of the leaf. But yes, this is it's a fascinating thing. I think it'll be. I think looking at the UV is certainly something that'll be really interesting and very straightforward to do.
You need to do some predation experiments.
Well, I know! You're exactly right, but try and work on endangered honey creepers.
Let's thank Rosie again for